Hi everyone, and welcome to today's Biology Daily Booster. This is number 18 in our Paper 2 series, and this is advanced information listed topics for both the foundation biology and higher biology. What we're going to start off with then is having a look at vaccines. So vaccination is one of those questions they do like on your exam paper. So first thing, what is a vaccine? It's something that contains a small amount of either dead or attenuated pathogens. Now, attenuated just means it's weakened. In order to get it into the body, we usually inject it. So if they ask you how do we administer or deliver a vaccine, injection is what they're looking for there. And then once that's in the body, because it is made of these pathogens, your lymphocytes, the white blood cells that make antibodies, are going to produce antibodies to that particular pathogen. What we're aiming for is that once we've made those antibodies, we're also going to generate these things called memory cells, which are going to remain in your body. So that means if you then to get exposed to that same pathogen in the future, then your memory cells replicate quickly, produce the antibodies quickly, and they can actually clear the infection before symptoms show. So that is what we mean by immunity. We're going to have a look at a few anti-words in our booster today that we need to make sure we've got straight in our heads and we're not mixing them up. So obviously antibodies produced by the lymphocytes, already looked at that one. Our next one is the antiseptic. Now antiseptics are what we use on external living surfaces in order to kill any types of pathogen that are present there. So this is the kind of stuff that we would use pre-surgery on the area we're about to cut open. So we use an antiseptic in order to kill any pathogens on the surface of the skin before we make that incision. So two good examples, alcohol and iodine. If we're thinking about non-living surfaces, so that would be like your kitchen counters for example, we use disinfectants. So antiseptics, living surfaces, disinfectants, non-living surfaces. Both kill pathogens. If we want to try to treat an infection caused by a virus, then we have to use antiviral drugs. So they will act on one particular type of virus, and the way that they're going to act and stop this infection varies depending on the antiviral we're looking at. So some will just block the virus from entering the host cell. They can't enter the host cell, it can't replicate. Others prevent it releasing the genetic material, obviously stops replication again. And some others will actually prevent the virus from inserting that genetic material into the host DNA. So all of them prevent virus replication, but just in a slightly different way. If we're dealing with a bacterial infection, it's antibiotics. So antibiotics kill bacteria. And again, we've got a range of types of antibiotic that work on a different type of bacteria. In order to actually work out the type of bacteria that's making someone ill, we'd actually have to take samples, grow them on agar plates, and then we would use these little antibiotic discs on an agar plate to work out which drug is best. This isn't what we do normally. If you're ill and the doctor thinks it's a bacterial infection, then they will just prescribe you an antibiotic that should work. They're not going to take samples in most cases. We only do that if there's a particular issue. Now this is the PAG that is listed on there for this particular unit. So looking at the effect of antimicrobials. The way that we're going to do this then is we start off with our little agar plate. So we've got our little plate with agar, and we're going to cover that with a lawn of bacteria. Now by that what we mean is we've spread bacteria across the entire surface of that agar plate. So if we were literally to stick that in an incubator, we would get a lovely coverage on the surface of just bacteria, so cover the whole surface there. Because we want to test antimicrobials, then this could be antibiotics, it could be obviously some other chemicals that are going to kill our bacteria. So what we tend to do here is we get these little tiny paper discs, that we're then going to soak in our relevant chemical, so whether it's antibiotics or some kind of antiseptic, for example, and then we place them onto the surface of that agar plate. The way we do that, we are going to use sterile forceps first of all, in order to lift up those little bits of paper. We obviously need to consider how we're going to make sure that they are not contaminated. We're going to sterilise them first. So sterilising the forceps could be hold them in the Bunsen flame first of all, let them cool off, and then you can lift your little paper disc up. We need to consider a control, so something that we can then compare to make sure it's not the paper. So the control in this case is literally just the little paper disc with nothing on it. So you would place the little paper disc with nothing on it as our control, just to prove it's not the paper causing the effect. Once we've got this, we're then going to place it in an incubator for a few days to give the bacteria a chance to grow. When we remove it, we end up with something like this. So this is showing us the effect of different antibiotics on our bacteria. And what we're really looking at is the thing called the zone of inhibition. So the region where bacterial growth has been prevented around that antibiotic disc. The larger the zone of inhibition, the greater the effect on killing the bacteria, because the clear zone means the bacteria have been killed. So if we have a look, whatever this one is down here, terrible. Doesn't look like it's killed much at all. Tiny little edge there, but not really. This one is by far the best. Now, if we wanted to prove that, we'd have to work out areas. So going to your maths knowledge, area of a circle, just pi r squared. Remember, radius is half the diameter. So use your ruler, measure the diameter of your actual circle, divide it by 2 to get your radius, obviously, and then square that, times it by pi, using the pi button on your calculator, and you've got your area of that particular zone of inhibition. Do that for each of them. The largest one is the best antibiotic to use. One of the things we need to be aware of when we're doing any practicals to do with microbiology is we need to do what's called aseptic technique. Now, the whole purpose of aseptic technique is it prevents any foreign microorganisms being introduced into our test sample. So we're making sure that the apparatus, the environment that we're actually doing this in are all sterile. So how do we do this? First thing is we're going to make sure there's no microorganisms present where we're working by washing down those working areas with alcohol before and after. Because if we're using microbial agents throughout this experiment, you also want to make sure they're not left there afterwards. To avoid contamination on our skin, we're going to wear gloves. Because we want to make sure that any of the apparatus we're using isn't going to contaminate our sample, we need to sterilise that. So we can autoclave this. So that's basically expose it to a high temperature to kill off all of these bacteria first of all. And to stop any microorganisms that are present in the air falling onto our agar plate, then we actually work close to a Bunsen flame. And that just means that it just keeps those bacteria away from actually falling into your dish. If we're considering how we're going to make what we refer to as a streak plate, then what we're going to be doing is using a little wire loop, and we're going to use an agar plate. So first thing we've got to do is we need to sterilise the wire loop. So to do that, what you do, hold it in the roaring Bunsen flame until it glows bright red, and then you move it just out of the flame so it will cool down. You don't wave it around in the air or move it far from the flame, because otherwise you're going to potentially introduce other microorganisms onto it. Once we've done that, we take the sample just by lifting it from the first agar plate or from dipping into the solution that we've got. And on our little agar plate, so this is our little plate, you then make 4 to 5 streaks along one edge. Flame the loop, let it cool again. You turn the plate ever so slightly, and then you're going to basically make those same little 4 to 5 streaks across the next edge. Flame the loop, let it cool, and then turn it, drag it down again, and so on, just until you're back to here. We don't make a complete square, you literally just go around leaving a gap between them. Place it in an incubator, And what's going to happen when it comes out is we're going to have quite a lot of bacteria in that initial area, getting a bit less the further round we go, until we're down to individual colonies at the end. Because each time we've actually dragged them across, what we've done is we've reduced the numbers, so we've spread them out, reducing the numbers so we can pick off individual colonies. That means we can identify them. Generally speaking, we look at them, so colours, the shape, whether they're raised, what the actual edge of them looks like, is it smooth, is it rough, and then we can carry out further chemical tests to identify it as well. The last part of our little booster today is going to be looking at how we get new drugs. So generally speaking, when we're talking about developing new drugs, these are going to come from plant extracts, or we may create them in the lab. But we are going to have to go through a range of different steps to make sure we've got a drug that is safe and effective at the end of the day. First thing we do is computer modelling. So we use a computer model to actually work out if the drug we're developing is likely to be effective, first of all. Once we've actually identified potential drugs to use, we're going to test it in the lab, in what we refer to as preclinical tests. Now, this is where we're going to expose some live cells, some bacteria, or a bit of tissue culture to our drug and see what happens. If obviously it kills all those cells or causes something a little bit untoward to happen, it's not going to go any further. If, however, everything looks good at that point, then we're going to move on to the next stage, which is animal testing. In order to pass the animal testing phase, we've got to pass 2 species before we can move on. To make animal testing a little bit more palatable, then what we use are our 3 R's principle. So reduction, use the smallest number of animals possible. Refinement, we improve experiments to avoid unnecessary suffering. And replacement, swapping out animals with our cell cultures, so that means we should obviously improve animal welfare in the long term, because we're not going to expose them to drugs that are incredibly harmful straight away. If we pass the animal trials, then we're going on to the clinical trials. Now, clinical trials are the ones involving humans. 3 stages to them. First one, healthy volunteers, and we're going to give them our drug to see if there's any side effects. Secondly, we then test a small sample of volunteers with our desired condition, and we're doing that to see how effective our new treatment is. If it all goes well there, we move on to stage 3, the large number of volunteers with our condition, and that's again just to test the effectiveness, check for safety in a large group. The last thing we need to be aware of is what we refer to as the placebo effect. So the placebo effect is where people start to feel better because they expect to feel better. By that I mean you've given them something, you've told them it's a medication that's going to make them feel much better, and because they believe that's what they're getting, their kind of mind over matter scenario happens and they do feel better, even though you've given them no chemicals that would actually help their condition whatsoever. In order to try to overcome this placebo effect, we use what we refer to as a double-blind trial. So in a double-blind trial, some patients will get the drug and others get a replica of it, so it looks the same, but there's no active ingredients, so they get the placebo. In our double-blind trial, neither the doctor carrying out this trial nor the patients receiving the drugs actually know who gets which. So that removes any element of bias here at all. It removes the placebo effect from the patients, and therefore we get more reliable data from these trials. If it's just a single blind trial, that's where the patients don't know, but the doctor does know who gets the medication. All fine, unless obviously the doctor has a vested interest in making sure this drug gets marketed, i.e. they're paid by the drugs company, in which case that bias could come into those results. As always, if there are bits you'd like to go over in a bit more detail, then you can use the video on the main channel. You can obviously use your revision guides, and I do recommend having a look at some of the OCR exam papers for Paper 2 Biology, just to get a bit of a feel for them before you go into the exam. And of course, don't forget to join us again tomorrow for our next Daily Booster.